So really quickly today, I want to talk about how once we've gotten a voltage change in the cell membrane to the terminal of the axon, how we're going to change that voltage into a release of neurotransmitters. So we've gotten this voltage change here, and we have these voltage-gated calcium channels, and so when they sense that change in voltage, they'll have a conformational change, which means they'll change shape in some way that allows the calcium into the cell. And when the calcium enters the cell, it will bind onto this protein called synaptotagmin, which is in a complex with snare proteins, which are holding on to this vesicle. It will cause a conformational change in the snare proteins, which brings this vesicle very, very close to the plasma membrane of the cell. And it will get close enough to where it can fuse. And when it fuses, it will exocytose. It'll spit out all of these neurotransmitters into the synapse. And from there, they can be sort of picked up by this sort of a receptor. And if the signal is strong enough, it will make it to the next axon and the next terminal. So really quickly, the voltage-gated calcium channel, it's calcium inside the cell, which causes conformational changes in the synaptotagmin and snare proteins, which cause a binding, a fusion of this sort of trapped vesicle with the plasma membrane.